Ever look in the mirror and see a bit of your mom's eyes and a bit of your dad's smile, but you're definitely not just a 50-50 split? Well, there's a reason for that. It all comes down to an incredible cellular process that shuffles the genetic deck. Today, we're diving deep into meiosis.
So, here's the game plan. We'll start with a puzzle, a big one, at the heart of reproduction. Then we'll look at the elegant two-part solution, meiosis 1 and meiosis 2. And finally, we'll zoom out and talk about why this whole process is so incredibly important for, well, pretty much all complex life.
Okay, first up, the chromosome conundrum. You see, when it comes to sexual reproduction, nature had to solve a really fundamental math problem to make everything work without going completely haywire.
I mean, think about it, right? If your mom gave you all of her chromosomes and your dad gave you all of his, you'd have twice as many as they do, and your kids would have 4 times as many. Very quickly, cells would just be bloated with DNA. So how does life avoid this genetic pileup? It all starts with having 2 different kinds of cells.
So, most of the cells that make you, your skin cells, your neurons, your muscle cells, are what we call diploid. All that means is they have 2 complete sets of chromosomes. You get 1 set from your mom, 1 from your dad, and we use the little shorthand 2N to represent that.
But for reproduction, you need something special. You need haploid cells. These guys have only a single set of chromosomes, no pairs. We call these 1N, and you probably know them by another name, gametes or sex cells. And this slide really lays out the difference nice and clear. Your normal body cells, the diploid ones, they use a process called mitosis to make identical copies. That's how you grow and heal. But to create those special haploid gametes, you need a totally different playbook. You need meiosis, and its whole job is to cut that chromosome number in half.
Which brings us to the main event. How on earth does a cell manage to cut its chromosome number perfectly in half and at the same time create brand new genetic combinations?
Here is the absolute golden rule of meiosis. If you take away just one thing, let it be this. In mitosis, the DNA copies itself once and the cell divides once. Simple. But in meiosis, the DNA still only copies itself once, but the cell goes through 2 rounds of division. That's the secret sauce.
So here's the basic blueprint. First, the cell bulks up and copies its DNA. That's interphase. But then it goes into the first division, meiosis 1, immediately followed by the second division, meiosis 2. So let's break down that first division, because that's where all the magic really happens. Welcome to meiosis 1. This is often called the reduction division, and for good reason. It's where the chromosome number actually gets cut in half. But it's also where the genetic deck gets a serious shuffle. Right out of the gate, something happens in meiosis 1 that never happens in mitosis. It's called synapsis. The chromosome you got from your mom finds its matching partner from your dad, and they pair up side by side. They get real cozy. And while they're all paired up like that, they do something incredible. Crossing over. They literally reach over, break off pieces of themselves, and swap them. It's like they're trading genetic information. This creates brand new hybrid chromosomes that are a unique mix of both your parents. And this is our first huge source of genetic variation. But wait, there's more. The second big source of variation happens next. When all these chromosome pairs line up in the middle of the cell, how they line up is totally random. For each pair, it's a 50-50 shot whether the mom chromosome or the dad chromosome faces left or right. It's like flipping 23 coins all at the same time.
So what does that coin flip get you? Well, with 23 pairs of chromosomes in a human cell, that random alignment, what we call independent assortment, creates over 8 million possible combinations of chromosomes. And that's before you even count the new gene mixes from crossing over.
It's mind-boggling.
Okay, now for the big split. During anaphase 1, the cell pulls the homologous pairs apart. This is so important. It's not splitting the X-shaped chromosomes down the middle. No, it's pulling the entire duplicated mom chromosome to one side and the entire duplicated dad chromosome to the other. So after that first division, what do we have? 2 cells. And here's the key. These cells are now officially haploid. They each have only 1 set of chromosomes. Now, each of those chromosomes is still made of 2 sister chromatids. They're still in that X shape, but the pairs are gone.
Which takes us to the grand finale, meiosis 2. And here's the good news. If you understand mitosis, this part is going to feel like a walk in the park. It's pretty much the same thing, just happening in a haploid cell this time. Yeah, there's no more DNA copying, no more complicated pairing or crossing over. The goal here is simple. Just separate the sister chromatids that are still stuck together.
And here we go. In anaphase 2, the cell machinery finally grabs onto the center of those X-shaped chromosomes and pulls the sister chromatids apart. Each one now becomes its own full-fledged chromosome. It's the exact same move you see in mitosis.
And scene. What's the final result? We started this whole journey with one diploid cell, and we've ended up with 4 haploid cells. And because of all that shuffling in meiosis 1, every single one of these 4 cells is genetically different from each other and from the cell that started it all. So, after all that, why does this matter? Why go through all this incredibly complex cellular gymnastics? Well, let's put it all into perspective by comparing meiosis to its cousin, mitosis.
This table really says it all. Mitosis is for making clones, identical diploid cells for growth. Meiosis is for making variety, 4 unique haploid cells for reproduction. And the key differences are right there in the middle. Mitosis has 2 divisions, and it has those game-changing events, synapses and crossing over, that you just don't see anywhere else.
So just to wrap our heads around it, the variation comes from 3 places. You have crossing over, mixing up genes on the same chromosome. You have independent assortment, shuffling which chromosomes end up in which cell. And then there's the 3rd level, random fertilization. Any one of those millions of unique sperm can meet any one of those millions of unique eggs. It's this incredible combination of factors that makes every individual on Earth, except for identical twins, totally unique.
And that leaves us with one last really big question to chew on. Meiosis is an astonishingly complex dance. It had to have evolved from the much simpler process of mitosis, but that meant adding all these wild new steps. How did life come up with such a brilliant, yet incredibly risky way to shuffle its own genes? Well, that's one of the great puzzles that biologists are still piecing together today.